Hi to all of you and welcome back to the series of lectures in molecular biology. This is the 14th lecture in which we will be discussing the translation introduction, specifically focusing on the definition, genetic code, and then different aspects of the translation. This lecture with the introduction to translation, as we already know, translation is the third step in the central dogma of unidirectional flow of genetic information, and it involves the conversion of information which is coded as the nucleic acid sequence, and this sequence is exactly translated to a specific amino acid sequence, resulting in the formation of a polypeptide. Simply, it can be identified as the protein synthesis as well. And this protein synthesis or translation involves a specific system which has a capacity to catalyze the linking of the amino acid called as the polymerization, so as to form a polypeptide sequence out of it. And it requires three things essentially, that is the mRNA, which carries the message of how to link the specific amino acid sequences, tRNA, which brings the amino acid to this system, and the rRNA, ribosomal RNA, which possesses the reading capacity of the mRNA, converts this sequence and links the amino acids together to form proteins. So all these three kinds of RNAs are present in this system, as you can see in the accompanying diagram. You will see the ribosomal rRNA is present as the green, and the tRNA here, which brings the amino acids bound to them to this complex, to this system, and the mRNA in itself, which is read from 5 prime 3 prime direction, so as to code for the polypeptide. This complex or this system is needed for the synthesis of proteins. So translation has to happen through a specific thing which is to be read, that's called as the mRNA, which carries the message of how to link different amino acids together to form a new polypeptide, or simply as protein. It's always read in a 5 prime 3 prime direction, that is the mRNA start is at 5 prime direction and it moves towards its 3 prime direction. The 5 prime start codon, or the start part of the mRNA, codes for the amino acid terminal of the polypeptide, and the carboxylate end of the polypeptide is coded by the 3 prime end of the mRNA. So we can in turn say that 5 prime end of the coding region of the mRNA contains the code for the amino end of the polypeptide. So 5 prime end will always represent the amino end of the polypeptide chain, and 3 prime end will always represent the carboxyl end of the polypeptide chain which is being synthesized. So the first amino acid which will be coded will be the one which is at amino end, and the last codon will code for the amino acid which is at the carboxyl end of this polypeptide. So in order to decode the information which is present in the mRNA, there should be certain language which should be understandable to the reading tRNA, as well as the catalyzing rRNA. So this universal language which is being read by the translation machinery is actually called as the genetic code. It is the linear sequence of the nucleotides within this DNA, and which is then transferred into the mRNA molecule, which has to be decoded by the tRNA anticodon arm. And usually it is present in the form of a triplet nucleotides, which are called as the codons, as you can see in the accompanying picture. And I have already told you that the mRNA is read from the 5 prime direction to the 3 prime direction. You will see the triplet codons. Let's for example say here A, and A. So these triplets make up a codon AAA, which codes for the lysine. Similarly, any other triplet codon will usually represent the amino acid. This is how the language is interpreted during the translation and the interpreter is the anticodon arm of the tRNA, which interacts with the codon or of the mRNA being translated or being decoded by the translational machinery. As we are already aware that there are 4 different kinds of the nucleotides present in the DNA as well as the RNA, and these are the 2 purines and pyrimidines. So 4 can give us the mathematical proposition of the 64 codons. That means that in the genetic code, the whole vocabulary is made up of 64 words, 64 codons, and these together constitute a genetic code which is to be decoded during the. Within this 64 different kinds of codons, only 61 are the ones which are representing any kind of an amino acid. That means that one codon is for this specific amino acid and codes for it during the polypeptide synthesis in the translation process. One of the codons of these 61 codons are coding for the specific kind of an initiator amino acid, which is methionine in eukaryotes and N-formyl methionine in prokaryotes. So methionine is decoded by the initiator codon, which is AUG, whereas in prokaryotes it is N-formyl methionine. So these are the ones which make up the amino acid or amino group of the amino terminal of the polypeptide chain finally. And you can see it here, AUG here. And there are 3 also codons which do not code for any kind of an amino acid, and these 3 codons are called as the stop codons. They code for the termination process of the translation. So termination of the translation is brought about by them. These are ochre, amber and opal, UAA, UAG and UGA, which you can see here, UAA, UAG and UGA. We are aware that general kind of an polypeptide contains 20 different kinds of amino acids. So out of the 61 codons which code for the amino acid, many codons are there which code for the same kind of an amino acid. So inversely, we can say that there are 3 amino acids, serine, arginine and leucine, which are coded by the 6 codons, as you can see here in the table, and there are 5 amino acids which are coded by the 4 codons. For each of the amino acid there are 4 codons each for them, and this process or this phenomenon is called as the degeneracy of the code. So when we say that the codon or the genetic code in the is usually having a degeneracy, we mean by that that there might be a codon which codes for more than one amino acid. Then there is another process or another thing which derives from this degeneracy, is that the third codon or the first base of the anticodon which decodes the codon, or the third base of the codon in itself, is not that too important. That's why there are certain codons which code for this same kind of an amino acid. This phenomenon is called as the redundancy of the third base, and because of this redundancy we can say that out of the 60 codon which needs the decoding process, only 30 to 35 fit the bill in decoding it. So we don't need 61 tRNAs to code for 61 codons. There are only half of them are needed for the decoding process to take place during the translation process, and rest of the amino acids, and you will see usually have 2 codons for them. Each amino acid has at least 2 codons, except methionine and tryptophan, which have one codon only, and rest usually will have a degeneracy and an ambiguity of the third base. Another thing which is important in the translation process is the Wobble hypothesis, which was given by the Crick, and it explains the ability of a tRNA molecule to recognize the multiple codons, as we already saw in the decoding of the genetic code. So the adapter molecule, which is a tRNA molecule, will have a ability to decode or to recognize many different codons. And this literally means that there is an interaction between a codon of mRNA and an anticodon of a tRNA is actually not specific. And the non-specificity is because of the two interactions, two non-standard hydrogen bonding interactions, which are Gu interactions and inosine interaction with A, U, and C. So the G not only will interact with the C as is complementary to the Watson-Crick model, but it will also interact with the U and vice versa, as well as the inosine. If it is present in the tRNA, then it is able to interact and base pair with the adenine, uracil, and cytosine as well. As you can see in non-specific interactions, let's take an example. For example, if an anticodon codon has an interaction of guanine and uracil, and if the scenario is that the tRNA anticodon has a sequence of 3' 5' XXG, then it will be two different codons having the sequence 5' 3' XXU, 5' 3' XXC. Similarly, for the U, it will be able to identify two different codons. Similarly, the interaction between the inosine and three different base pairs have been identified, and if the anticodon arm is having a sequence of XXI, then it will be able to identify three different codons having A, U, and C at a complementary position to that of the I. As you can see in the accompanying picture here, if the anticodon of the tRNA is like this, then it will be able to read the mRNA of three different codons within this mRNA. And the same kind of a scenario is given in different way in this kind of a picture, where the anticodon codon interaction is for the one codon reading only, and if the anticodon is having a U, then two codons, and if it is having I, then the three codons will be identified by this kind of an tRNA. This is what Wobble hypothesis means. That means if the third position of the anticodon has a G or I, it will be able to wobble, it will be able to read more than one codon. So in humans, the GU interactions are known to be used for the 8 tRNAs, and in every case, the sequence of a tRNA anticodon is XXG, and anticodon with the sequence of XXU are not utilized in humans because of the very weak interactions between a codon and an anticodon. And also we know that in humans there are T8 tRNAs which have anticodon sequence of XXI. But instead of utilizing this for the recognition of three different codons, it decodes only XXU and XXC codons. Further moving to the aspects of translation, one of the important aspect of the translation process is that the requirement of a certain machinery, which we know now is the ribosomes. Ribosomes act as sites or the seeds for the decoding of the information in mRNA molecule, which is to be put as the sequence of the amino acid to synthesize the polypeptide. And in order to decode the information in the mRNA, the prokaryotes have a specific three different sites for decoding the information and carrying out the translation process. All these three sites are the function of the two different subunits of the prokaryotes, the 50S subunit as well as the 30S subunit. And there are three sites present in the prokaryotes which are characterized as A site, which is the amino acid site, and then there is a P site, and then there is a E site. A site or amino acid site is the one which accepts the amino acid tRNAs, the tRNA which are charged with the amino acids, to its site during the elongation phase. However, what happens in the initiation phase, the first codon is present at the P site, peptidyl site, and the first charged tRNA with the F-met formyl methionine, charged with the formyl methionine, this tRNA binds to the initiation codon during the synthesis of the tRNA. So the first tRNA which binds during the translation process always binds to the P site, and then later on all other succeeding amino acids will come and bind to the A site, then they will move to the peptidyl site during the synthesis process. And finally is the exit site, E site, which is present in the 50s subunit of the prokaryotes. This site is not present in the eukaryotes. Eukaryotes carry only A and P site. One of the important factors which regulates the translation process is that the translation start site is defined by the conserved sequences, similar to that of the promoter regions of the mRNA. Here also, the conserved sequences are located upstream of the first codon to be translated. So if the start codon is like here in the picture, you will see there is a certain untranslated region which is present in all kinds of an mRNA, and this untranslated region contains certain specific sequences which are highly conserved, usually 18 to 30 base pairs. These are called as the Shine-Dalgarno sequence or Sheen-Dalgarno sequence, having a sequence of conservation which is AGG-AGGU. You can see the conserved sequences here, and this conserved sequence is actually complementary to the specific sequence which is present in the smaller subunit of the ribosomes, present usually in the 16 sRNA at the 3' end, and these are needed for the interactions to take place between them so that the 30s subunit will bind to the mRNA which needs to be decoded, hence enabling the initiation of the translation. Second aspect of the translation process is also that it is not always that only a single ribosome will bind to a single mRNA. Actually, what happens is that the mRNA molecule is bound by numerous kinds of clusters of the ribosomes, usually 10 to 100. When this happens, they form beads in a string kind of a structure. Those are called as the polyribosomes. So essentially this is the mRNA which is read from 5' 3' interaction. You will see a number of ribosomes are bound to this single mRNA run and making up as polysomes, and in turn you will see that each of the ribosomes are synthesizing a polypeptide. So actually a single mRNA decodes numerous kinds of polypeptides in one go. So out of one mRNA we get a number of polypeptides of a single protein at one time simultaneously. Actual translation process can take place. We need to decode the codons of the mRNA during the translation process, and for this we have tRNA, the anticodon of the tRNA molecule, which acts as an adapter molecule, interacts with the codons and decode the information so as it will incorporate the sequence of amino acids as directed by the codon sequences in the mRNA molecule during this translation process. And this adapter molecule needs to be charged with these kinds of amino acids. Particularly, we know that only 20 different amino acids are present in the universally in the nature, and there are around 61 codons which need to be decoded for these 20 amino acids. In prokaryotes, there are 30 to 45 different tRNAs for doing this function of decoding the codons. While as in eukaryotes, because of the complexity and because of the increase in efficiency, 50 tRNAs are existing for decoding purposes. We know that out of this probability that there are 50 different tRNAs or 30 to 45 different kind of tRNAs to code only just 20 amino acids, so there is a probability that there are certain tRNAs which are linked with the same kind of an amino acid. These tRNAs which carry the similar or same amino acids are called as iso-accepting tRNAs. The amino acid which binds to the tRNA binds it to a specific site. Those are called as amino acid attachment or amino acid acceptor arm of the tRNA, while as the another arm is called as the anticodon arm, which codes or decodes the information while interacting with the codon of mRNA, anticodon-codon interaction. So the attachment which happens at a amino acid acceptor arm at a sequence of CCA, and the tRNA which accepts the amino acids is called as charged tRNA. The charging or the amino acylation is brought about by specific enzymes. Those are amino acyl tRNA synthetases, and those are of two different types. Two types of an enzyme depends upon which hydroxyl group in the adenosine moiety of the CCA arm will be charged with the amino acid. If it is type 1 enzyme, it will charge the amino group 2 and link it to the 2' hydroxyl group of the adenosine moiety in the CCA group. If it is type 2, then the amino acid will be transferred to the 3' hydroxyl group of the adenosine in this arm CCA of the tRNA molecule. However, the kinetics of the both remain the same. In the kinetics is a two-step process. In the first step, the carboxyl group of the amino acid reacts with the alpha phosphate of the ATP and makes an anhydride linkage, which in turn produces the enzyme bound intermediate, which is called as amino acyl adenylate. When the amino acyl adenylate is formed, this sort of charges the amino acid for the further down reaction. So what happens here is that the amino acid group is charged with the ATP to form the enzyme bound complex of amino acyl AMP, and then this amino acid group is then transferred to the either 2' hydroxyl group or 3' hydroxyl group, depending upon the type of an enzyme of the adenosine, and hence the tRNA is called as the charged tRNA. So the charged tRNA differs from the uncharged tRNA in binding of the amino acid to the 3' CCA arm. So the charging involves the utilization of the 2 ATP energy equivalent to 2 ATPs, because during the charging of this, the amino acid first formed the adenylate moiety, and then it releases the pyrophosphate. Pyrophosphate in turn formed the phosphates. So actually the energy of two phosphoanhydride bonds is utilized in charging the tRNA molecule. So for creating a later on creating a single peptide bond, two ATPs have been utilized up to this point. So for charging of the tRNA, the energy equivalent of 2 ATPs is utilized. To the another aspect of translation is the open reading frame. The concept of reading frame is actually the run of nucleotides which can be read before a termination codon will terminate it, and is usually referred to as open reading frame. So open reading frame is the run of the nucleotides within the gene or within the sequence of an mRNA which can be read. And as we know, in the random sequence of the nucleotides, one every 20 codons in the reading frame is on average a termination codon. So if an example of a mRNA is given, we can read it in three different ways. So three different possibilities are there, and three different polypeptides can be generated from a single run of mRNA. However, this is not possible in nature because of the availability of a start codon. That's why the start codons are important, because they actually tell the reading translational machinery, that is the ribosomes as well as the tRNAs, to read only one out of the three possibilities. So AUG starts the translation process, and then the triplets will follow the suit. So there is a possibility within a gene only to synthesize only one kind of an polypeptide. This is usually true for most of the prokaryotes and eukaryotes, except certain prokaryotes which have a limited genome. So there the possibility is that of gene overlapping another gene. So a part of a gene is a part of some other gene, also coding for some other protein also in those cases. So in this lecture, we tried to cover most of the aspects and introductions about the translation. And in the next lecture, I will be discussing the process of translation in itself. If you like the contents of the video, you can order the book Molecular Biology and Biotechniques from Amazon. It's available both in Kindle and paperback format. And do subscribe to my channel for further videos which I will upload in due time.